Okay, are y'all ready to get started? So, Dr. Wong asked me to give a guest lecture today on brain-machine interfaces. So this will kind of be like in high school when you had a substitute teacher and you just watched a bunch of videos, except it'll be a lot more interesting, I hope. So if you would be so kind as to give me your attention for about half an hour, 45 minutes or so, and I'm going to show you some things that I find really fascinating, and hopefully you will too, and then we can talk about them, discuss, you can ask questions, that sort of thing. So, in case you don't know me, my name is Luke, I'm TA. Hopefully you've seen me before. So I figured we could start by, I could give you a brief history of upper limb prostheses. This is the kind of work that I'm interested in, and so obviously I like to talk about it, so hopefully you'll be able to listen to it some. And then we can talk about brain-machine interfaces, what they are, what they do, how they work, things like that. And then we can look a little bit about how we can use that to restore limb function. So a long time ago, if you remember from your world history classes in high school, there were the Punic Wars. And you saw the very first documented prosthetic limb showed up around 200 BC, and it came from a Roman general during the Second Punic Wars, and he had a passive hand that he used to hold his shield. And then about 1800 years later, there were more wars, the Italian Wars, and there was another invention. It's called the Berlichingen hand, and it was also a passive hand, meaning it didn't move. But this was an Italian general who had lost his arm in battle and wanted to keep fighting, so he had someone make an iron hand, and so they called him the Iron Hand Man, I guess. Several years later, there was a French surgeon, who started to design limbs because there were a lot of soldiers who were losing them during these wars. And then the Napoleonic Wars, early 1800s, is when you saw the first, what's called a body-powered prosthesis. So this is a device that an amputee could wear and then actually power to move the limb or grab an object. And then more recently, World War I and II saw the development of the very first myoelectric prosthesis. Now, if y'all remember from your biointerfacing lab just the other day, you were using a myoband and collecting EMG signals. So this was one of the very first limbs to actually utilize EMG signals to make the hand move, open and close, things like that. What's interesting to note though is whenever there's a war, there seems to be an advancement in prosthesis technology. So most recently we had Operation Iraqi Freedom, Desert Storm and things like that, and there was an explosion of really sophisticated upper limb prostheses that hit the market. And so most notably, y'all are probably familiar with the modular prosthetic limb. This has come out of the Hopkins Applied Physics Lab. And y'all probably worked with Dr. Wester and Mr. Arminger during the labs. They actually are the guys that are the experts on this project. So y'all remember this, right? Y'all know what this is? Let's see. Well, oh well, there's sound, whatever. George Lucas had this crazy idea that, well, you know, when Luke Skywalker lost his hand, he just got a brand new one and it worked really well, and there didn't seem to be an issue. If you would, I'm sorry guys, I actually forgot to start this recording. Good thing we have a camcorder in the back. All right. Okay. So wouldn't it be great if we could if somebody lost their limb, they could just get a new one like Luke Skywalker did? And that's almost what the Applied Physics Lab modular prosthetic limb is doing, in a sense. It's probably the most sophisticated prosthetic limb out there today. It has 26 degrees of freedom and over 100, and I think it has 109 sensors throughout the limb. So there's a bunch of force sensors at the fingertips, accelerometers, things like that. So it has a very good sense about where it is in space, and it can actually mimic any human gesture that you throw at it. So it's one of the most sophisticated limbs today. And what's even cooler is that it's happening right here at the Applied Physics Lab just down the road. So let's move on to what an actual brain-machine interface is and what it does. So a brain-machine interface accesses brain signals and then uses them to achieve some level of control over a machine or a computer. So what we're going to talk a lot about today are prosthetic limbs. And these machines are built on the idea that you have information, sensory, motor, and some cognitive information that's being represented in your brain by your neurons. And so the idea is, well, maybe you can extract that information and use it to control something like a prosthetic limb. So this is a flow diagram of what a system might typically look like. You have some signal acquisition, you have some way that you're capturing the signals from the brain, and you digitize it, and you look at the signal, you do some processing. So in your lab, you probably did some pattern recognition. Maybe you were looking for specific EMG patterns or features to make a particular movement. You could do something similar with these neural signals. And then you have some algorithm that takes that information and issues a command. So perhaps it's typing out letters on a screen, or controlling a wheelchair, maybe even grabbing an object. So in 2006, the Defense Agency, sorry, let me get this right. The Defense Advanced Research Projects Agency, DARPA, had this project called Revolutionizing Prosthetics in 2006. And the goal was to significantly push the current technology for upper limb prostheses. Lower limb devices are pretty sophisticated, and upper limbs seem to be lagging behind. So in the early 2000s, they threw out a lot of money to these organizations to try to improve on the upper limb prosthesis technology. And the idea is, well, maybe, let's see if we can play this again. You want to be able to have somebody intuitively think about controlling a prosthetic limb, perform some action, and then get some type of feedback from that action. And the goal is to try to replicate or replace some lost functionality. Okay, so this picture comes from a review paper by Dr. Takor, who is here at Johns Hopkins. And he talks about the different types of signals you can get from the brain. And so you have things that are called local field potentials. And you can, if you stick an array of microelectrode needles in the brain, you can actually measure a field potential, an electrical potential from a population of neurons. Similarly, you could also use those electrodes to record single spikes from the brain. You could also take EEG sensors and place them on the scalp on the outside, so you don't actually have to go into the brain, and you can collect signals there. But they tend to have a slightly lower frequency, and that's because as it passes through your skull, it's essentially acting as a low pass filter, meaning some of your signals are getting attenuated, so you're left with a smaller piece of the entire picture. And then finally, you can have what's called ECOG, so electrocorticogram, where you have surface electrodes, similar to the electrodes that were in the Myo armband that y'all used, but these electrodes are made specifically to be placed on the surface of the brain. And so from these, you can also measure neural activity. Okay? So far, so good? All right. So let's look at these two first, EEG and spiking activity. So the first generation of BMIs used EEG signals because they were easy to get, they were non-invasive, you didn't have to cut anybody open to record from their brain. And the thought was, well, all right, let's collect these signals, signals and do some processing and then try to extract some feature. And then we're going to use that to classify a prosthetic hand as being opened or closed using only your brain signals. And so what they do is they have people come in, they put these EEG electrodes on their head, and they have them think of, say, opening this prosthetic hand and closing it. And then they're recording these signals while they're thinking this, and they're imagining these movements. And what you actually see is a change in brain activity based on what they're thinking. And so now you can use that difference in the signal to actually tell the prosthetic limb to open or close. So here's a video that came out of our lab a few years ago where this person is presented a cue and then the word open comes up. And so then he has to think about opening the prosthetic hand. And then now he's going to think about closing it. So he's using only his EEG signal to make this movement. And what's happening is you're recording the signal and processing it and then sending a command to the prosthetic hand to either open or close based on the type of signal that it's receiving. So this is another example using a slightly more sophisticated hand, but it is also using EEG signals. And what you're going to see here is a screen with the signals from this user's brain going across and causing the hand to open and close. Okay, now if you remember, Dr. Jeng in his lecture a few days ago showed you this example of spiking activity from the motor cortex. Y'all remember that? This actually comes from a group in Pittsburgh who has done single unit recordings and used that to control the prosthetic limb. So the video y'all saw of the monkey feeding himself, this is what's going on right here. So they put microelectrode arrays into the motor cortex of a monkey. And then they were measuring single spikes. And since y'all paid attention in Dr. Jeng's lecture, you remember that they used population vector to decode movements. And so this is a plot showing the type of neural activity that happens whenever the monkey makes a movement in a different direction. And so you can use things like population vector decoding to figure out what kind of movement the monkey intends to make whenever you see a specific pattern of neural activity. And you can use this to control the prosthetic limb to feed yourself. Now this is work that happened here at Hopkins as well, where we're using single spikes to control individual fingers of a prosthetic limb. So this is what you would call offline decoding though, because the monkey isn't actually attached to the prosthetic limb at the moment. And I'm sorry the sound's not working, but it is actually playing music. Let me show you again. So these signals are being fed into the controller of the prosthesis, and depending on the pattern that it sees, it's going to move different fingers. Pretty fascinating, huh? So this was some of the early work done for brain-machine interfaces, and then as time went on, people started getting a little more creative and looking at other types of signals. So I'm going to show you another example of using spikes to decode movement, but then ECOG also started getting popular because what happened is people with epilepsy come into neurologists, and a lot of times they will perform a surgery and put these electrodes on the surface of the brain to try to localize where a seizure is coming from. But what's happening is these patients will come in and stay in the hospital for about a week while they sit around waiting to have a seizure. And so there's been a larger opportunity to collect brain surface activity from these patients while they're sitting in the hospital. And so, well, let me show you this example of spiking activity first. So now this is moving more towards the future, the second generation of BMI. And again, this is the same group from Pittsburgh, the one that showed the monkey feeding himself with the spikes. And so they're still using spikes, but this time they put it in a human, okay? And so this is a huge step for brain-machine interfaces, because now there's actually a human with electrodes implanted in her brain, and she can use that to control a prosthetic limb. So one question for you is, well, here's the central sulcus right here. And these two black diamonds are very small microelectrode arrays. And so why did they put them there if they want to control the movement of a prosthetic limb? Any thoughts? Say it louder. Y'all know the answer. It's because this is the primary motor cortex that sits a little in the front of the central sulcus. And so whenever you make movements, you see a lot of activity in your primary motor cortex. And so the thought is, well, if maybe you can tap into that and actually record intended movements and use that to control the prosthetic limb. So here's what they did. They put these two tiny electrode arrays in the patient's brain, and they trained her on how to think about moving a prosthetic limb in three-dimensional space. So all of a sudden she's achieving very complicated movements using brain signals, just her thoughts, okay? So this is a video of what happened after she was training for several weeks. She hadn't been able to feed herself in maybe 10 years. And so for the first time in about a decade, she was able to think about controlling a prosthetic limb, to grab food and eat chocolate. And so this was a huge advancement in BMI, because it was a demonstration of humans actually using implanted microelectrode arrays to control prosthetic limb. Fascinating, right? And again, this happened at the University of Pittsburgh. Yes?
Can we go back to the previous slide?
Sure.
So, you may mention this, but why is the activity right lateralized in the brain?
Right here?
Yeah.
It could be a bunch of reasons. Maybe, so this was I guess an fMRI scan of her brain during some movement. And maybe, so it looks like, what would this be, kind of the temporal region? So maybe there's some auditory artifacts going on. I don't know, I could look closer and we could talk about it. But I mean, if you think about it, there's a lot of stuff going on in your brain at any given time, right? So it's incredible to think that you can pick out specific signals from the motor cortex and use that to move the limb around in space. Okay, so those are microelectrode arrays. Now let's talk about ECOG signals. So these are using electrodes that are placed on the surface of the brain. And what's really cool about this is that it's happening here at Hopkins. So what you do is you perform what's called a craniotomy, where you open up part of the skull and you place these electrodes on the surface of the brain. Well, I guess you and I wouldn't do it, right? You have to be a trained surgeon to do this, obviously. But then you now have this wide coverage of the brain, and you can measure a lot of different types of activity. One issue is, though, you don't get quite as specific information from individual neurons, but you have a large area of coverage. And what you can do, though, is when somebody's performing a task with these electrodes implanted, you can measure the activity at each electrode and use that information to figure out maybe how somebody wants to move or something like that. So this is a video from one of my lab mates who has used ECOG to control the MPL. Sorry, let me rephrase that. My lab mate isn't controlling it himself. There's a patient with epilepsy who's been in the hospital and has these electrodes implanted. But what we've done is come up with algorithms that take that information from the brain and use it to figure out, well, how is the limb going to move in space? Okay, and so most recently, just a couple months ago actually, they achieved individual finger movement from an ECOG array. Now this is pretty fascinating because you don't always get really precise electrode locations whenever you implant these grids, and so it's hard to really pick out fine features sometimes. And so what they noticed though is they were able to discriminate between the 5 different fingers on somebody's hand. And since you all remember the homunculus that you've seen a bajillion times by now, you know that the hands have a pretty large representation in your motor cortex, right? But you'll also notice, if you look closely, so this green line is the central sulcus. The electrode array covers both in front and behind the central sulcus. So what does that mean? Any thoughts? You get sensory information too, because you're spanning both the motor cortex and the somatosensory cortex. And what they found, so the experiment that they did was they would tap on the individual fingers to see which regions of the brain would light up. And then they would say, okay, well, when this region of the brain lights up, that's telling me something's going on at this finger, or maybe my pinky, or maybe my thumb. And if you notice, you have a lot of sensory representation on this side and motor representation over here, but it's all communicating with each other. So if I tap on my finger, I feel something, so it's going to activate my somatosensory cortex, but then at the same time my finger is moving when you press on it, so that's also activating my motor cortex. So you can take that combination of activity to figure out what's going on. Yes?
So is all of this very specific to a person? Like you have to run all the tests to try to figure out for that specific person? Like
Sure, yeah. So it varies by person, but this in general holds true. The biggest factor though is when you implant these electrodes on somebody, they're not always going to be exactly where you think they are every time. So maybe they get implanted and then over time they move, or maybe there's some buildup that causes the electrodes to fail. So it is different per person, but in general this is true. So if you're on Facebook, you know what IFL Science is, and they picked this up real quick. So this is Guy Hodson, my lab mate, who published this paper, and it's pretty cool. And I'm going to show you a video of it in action. Let's see. So what's happening here is the patient in the bed is going to see a cue on the screen, and that screen is going to indicate which finger he's going to move. And then the prosthetic limb here is going to take the activity from his brain and try to figure out which finger he's moving, okay? So let's see how it does. Oops. Now, this might not seem impressive, right? Because you think, oh, well, he's moving his finger and it's kind of slow. But keep in mind that this is responding from only the brain signals from this person. Yes?
Is there a reason he's like pulsing his fingers as opposed to pulling them?
I think it was just the way the experiment was set up, was to keep moving. Because your brain responds a lot to change. So maybe it had something to do with constantly changing your finger position. So like, for instance, you remember rapidly adapting mechanoreceptors in your somatosensory system. They activate when you initially press them, but over time they stop activating. So in general, things respond more to changes, and so that could also be why they wanted him to keep moving it. Okay, So far, so good. We've been talking a lot about brain signals, right? And how you can control a limb with your brain. But I'm going to talk a little bit now about EMG control, okay? And so this is what y'all were doing in your lab the other day. You were collecting EMG signals and using that to control the virtual prosthetic limb, is that right? Yep, okay. So there's this surgery called targeted muscle reinnervation. So let's take a step back and think what happens when somebody has an amputation. Right? They lose part of their limb as well as some of the surrounding muscle, okay? So they no longer have all the muscles to control a prosthetic limb, for instance, okay? So imagine you have a keyboard and you need to type every single letter to spell out words, but you only have 2 keys. That's a common issue that a lot of upper limb amputees face, where they have an amputation, but they only have 2 EMG sites that really produce a lot of activity that you can discriminate against. And so it's like as if you only had 2 keys on your keyboard and you had to type out combinations of both of them to spell out an entire word. So that led to this idea of targeted muscle reinnervation. Well, what if you take a large muscle like your pec and cut it into 3 different pieces, and if you place your 3 major nerves that are running down your arm, your median nerve, your radial nerve, and your ulnar nerve, I guess this is musculocutaneous nerve as well. You have multiple nerves, right? But the idea is you cut your pec into different sections and then place the nerve into those muscles. So what happens is the nerve regrows and attaches itself to those muscles. So now you can actually move your pec independently, these 3 regions independently. So now you have more inputs to control the prosthetic limb. Does that make sense? Okay. So y'all probably did some of this in the lab, right? You collect your EMG signal and you have some, say, vector, some matrix of weights telling you, or activity telling you how important each channel is to the particular thing you're trying to do. So for instance, if I'm trying to close my hand, I have more activity maybe on this side of my arm than over here. If I'm pointing my finger, maybe all of my activity is up here on the top of my forearm. And so you can take those signals and run some classification to do pattern recognition. And the idea is you have different groups of EMG activity that correspond to different hand movements. And so this is what some of the data might look like, where on day 1, say you have different cues. You open your hand, you close your hand, you try to do an index point and a fine pinch, and all of your EMG activity is kind of jumbled up together and it's hard to separate it out. And so with training, you can actually begin to distinguish a little bit better between these different movements. And so that's the idea behind pattern recognition. Okay, so this is an example of a gentleman who lost both of his arms when he was young from an accident, and he had what's called a shoulder disarticulation. So he has no shoulder movement. He has his pec muscle, but he doesn't have shoulder. And so this is a long video, so I'm going to try to find the interesting part. Okay. If you don't recognize him, this is Dr. Albert Chi from Hopkins, and he's actually the one who performs these surgeries here. And so Les, the amputee, is being fitted with a vest that's going to contain a bunch of electrodes that are being placed on different parts of his chest. So they had this targeted reinnervation surgery where they cut his pec into multiple pieces and then put nerves in the different sections. And so the thought is, well, now he can control this really complicated limb that you saw in the beginning, could make all of these sophisticated movements using only his muscle activity. In a sense, he thinks about how he wants to move it, and that's activating different parts of his muscle, and that's being picked up by the MPL, and then that's causing it to move. So what you see here is he's training in that virtual environment that y'all probably used the other day. And this is at APL. So he trains, you collect all this EMG information to build your classifier, and then you're able to use that to actually control the limb. So let's see. Here we go. So they're telling him to do different things, and then he's making the movements to actually move the limb. You can watch, so there's Mr. Arminger. You can watch this later if you want to with sound, but it's pretty fascinating to hear what he has to say and about how he feels when he's able to do these things that he hasn't been able to do for a long, long time. And what's even more impressive is this is the first time in history that a double amputee has controlled 2 MPLs simultaneously. Okay, in the last few minutes, let's now talk about sensory neural prostheses, because everything so far has been motor, right? You implant something in the motor cortex and you can pick up activity about how you're moving. Well, what about sensory information? What if you wanted to actually feel what you're touching or how you're moving in space? Because what you've been doing now is recording from your motor cortex and causing the robotic arm to move. But now let's see if you can actually use some sensor information from the arm and then convert that to some type of electrical stimulation to then give sensory feedback back to the user. So here's an example of a really incredible paper published in 2013 about stimulation in the somatosensory cortex. And this is a non-human primate, and they used microelectrodes and put it in S1. And what they did was they got a monkey and they tapped its fingers sometimes, and then they stimulated its brain in the region that corresponded to that finger sometimes. And so they performed essentially a psychometric function, if you remember way back from the first exam. You can use this to kind of differentiate, well, what's going on, what's the threshold for the sensation. And what they do is they train the monkey to make some eye saccade based on what it thinks it feels. So if it thinks you're touching its finger, it's going to look one way. And what they found is that it didn't matter if they actually touched his finger or if they stimulated certain parts of his brain, it felt like the same thing to the monkey. And that's what you see here with these 2 psychometric curves, where when they were pressing on his native finger, he has this curve, and then when they were stimulating his brain, he couldn't really tell the difference. He thought they were the same, essentially. So this is a really impressive example of improvements in sensory feedback. Okay, so what if you didn't have to open up the brain to control the prosthetic limb? You know, what if you could have some interface to the peripheral nerves and the muscles to collect EMG signals, and then maybe for people who still have intact spinal pathways, they then have these same sensations in S1 and M1. And then can you combine what's been done with the motor decoding with the sensory feedback to really restore limb function? And so that's some of the work I'm trying to do is, can you measure some touch signal on a prosthetic hand and then use that to improve how the hand is actually behaving? So this is a video of a prosthetic hand. There's no human attached to it, but it has no sense of what's going on. It doesn't know what it's touching. It just, you tell it to close and it closes. And so the thought was, well, maybe we can add some sensors to it to make it a little bit smarter, so then when it tries to grab the egg, it'll stop, because it knows how hard it's touching the object, and it can tell that, well, I don't want to press any harder because then I'm going to break it. So what we did is we had patients come in and try to perform tasks where they picked up delicate objects and moved them without breaking them. And what we recorded how quickly they could move and how well they were able to move it without breaking it. And what we found was it takes a lot of concentration and precision to really make those movements so the cracker doesn't break. But then what we did is we put sensors on the prosthetic hand, and it was communicating with the controller and telling the hand, well, hold up, you're grabbing too hard, or maybe you're not grabbing hard enough, and so the object slips. And then with this added functionality, we found that the users tended to get faster and they broke less items as they moved. Okay, and now what I just showed you though was without that sensory information wasn't being sent back to the patient, it was just going to the limb. So a couple years ago though, there's a group in Italy who actually put electrodes in the peripheral nerves and stimulated the nerves, and the amputee was blindfolded. And what they were trying to do was to see if he could discriminate between different shapes of objects and different stiffnesses of those objects. And so did y'all plot a confusion matrix in the lab last week, right? That's what they did. They said, okay, well, how good is he at figuring out if the object is hard, medium, or soft stiffness? And so what happened is when he grabbed the object, he would receive stimulation that corresponded to how stiff that object was, or essentially how hard he was grabbing the object. So if you're grabbing the object really hard, you're going to get a little more stimulation. And they did the same thing with shape. Is the object small? I think SP is square and CY was a cylinder. Oh no, sphere, sorry. Was it a small object? Was it a sphere, or is it a cylinder? And keep in mind the user is blindfolded, and so all he is going off of is the sensory information he's receiving back from the stimulation. But he's still controlling the prosthetic limb with EMG. So this is one of the first examples of a completely closed loop neural prosthesis, where he was sending both motor information to move the limb as well as receiving sensory information to feel what he was touching. For the sake of time, I'm not going to show this video, but it's a nice overview of the entire experiment that they did. So if you have some time, the link is down here. You should go watch it. Okay. Now the last thing I want to show you is really, really cool. This actually happened a couple weeks ago. This goes back more to the brain-machine interfaces that we were talking about earlier, but Nature just had a publication that was the first person who had quadriplegia. So he had a spinal cord injury, he was in a car accident, and he couldn't move his arms or his legs. But what they did was they attached stimulators to his forearm, and they're also recording activity in his brain, okay? So they're actually now going to try to move the human with these motor signals. So before they were moving a prosthetic limb to, they were using the motor signals to control the prosthetic limb, but now they're actually trying to move the patient. Here we go. Oh, shucks. This was the last slide I had, actually. So what I'm going to do is actually just pull up the link, if that's okay, and we could watch it on there, and then be happy to take any questions y'all might have. All right, so what he's doing now is talking about his accident and how he hasn't been able to move in a long time. Okay. But if you believe me, what they're doing is those gold bands that they were wrapping on his arm were sending electrical signals directly to the muscles. So he still had a healthy brain, right? He could still think about making these movements. So all they had to do was capture those signals, but then bypass his spinal cord and send it directly to his muscles. So here he's given a cue and he's thinking about making that movement. And then from those thoughts, they're taking that and stimulating his forearm. So again, this is kind of silly without sound. Sorry about that. But I know we still have a few minutes, and this is pretty much all I had. So if there are any general comments or questions y'all might have, shoot, now's the time to ask. Oh, before you ask, let's watch this. So this is before he had any stimulation. He had a really hard time controlling his wrist movements. But now what's happening is they're actually stimulating with this wrap, and it's causing his hand to move. So I guess I misspoke when I said he couldn't move his arms. He can still move his upper body, but he can't rotate and move around his wrist. And yeah, he's talking about how great it is. Okay, any questions? Yeah. Yeah, absolutely. You do, yeah, you run the risk of let's make sure. That's a good point, right? You run the risk of damaging your hand if you can't feel. You know, like sometimes you go to the dentist and you get numbed up and you bite your tongue and you don't know it until the pain medicine goes away and it really hurts. Yeah, so that is a potential problem. So maybe the next step then would be, well, because that connection to the spinal cord, through a spinal cord is broken, what if you add back in that sensory information that you saw in other examples? Yeah. And after a while, I imagine the system sort of implanted. Eventually, yeah. So as far as I know, the longest time somebody's been implanted is about 5.5 years with a microelectrode array. There are companies that specialize in making these. So they've done a good job at sealing them so that your body doesn't completely reject them. But for a lot of the ECOG patients, those are typically implanted for about a week. So you kind of mitigate that long-term effect. So that's one of the future challenges, is how do you design these systems to be long-term? You know, can you pull it off? Anybody else? Yeah.
So they've made progress with giving sensory feedback directly to the brain, where you're actually stimulating the neurons. That seems kind of high risk relative to just stimulating neurons in the arm. So those neurons control the muscles. So I'm curious if there's any efforts towards, now that you've actually got feedback going into neurons here, are there any efforts towards putting feedback into neurons here so that the neurons themselves control the muscles?
Absolutely. Well, so the feedback to the nerve, you have, when your nerve, you have sensory fibers, you know, and motor fibers. But what they're trying to do now is stimulate those afferent fibers, the fibers going back to the brain, to send the sensory information, so that you don't actually have to go into your brain to stimulate that spot. So a lot of times, like, you can still think about making a movement, right, which triggers efferent activity. The muscle fibers will go, but if you don't have any feedback coming in, you know, it's a little wacky. So there's been a big push recently. Actually, DARPA has thrown a lot more money out a couple years ago at exactly that. Can you come up with a peripheral nerve interface that will be safe, that you can use to stimulate and send sensory information back, so you don't have to go into the brain?
There's just one more question I had.
Sure.
So prosthetics, very helpful with like amputated limbs. But then there's also been a lot of progress made with restoring nerves, like through nerve growth and pathways to actually get them to regrow themselves. Do you see like efforts to, you know, control muscles with these outside bands that are increasing as you know, progress is being made with restoring nerve function for growth?
Sure. Maybe this is naive, but I would think they're complementary, because both are still pretty young in terms of the progress that's been made. I mean, this is the kind of stuff that you probably won't see people using walking around in the streets for another, you know, few decades. So I think at the moment, I would assume they would kind of grow in tandem, And then I don't have any insight as to which one might be better at the moment. Alrighty, well, if there's nothing else, y'all are free to go. Thanks for listening. Feel free to come talk to me if you're interested. We do a lot of cool stuff, and I'd be happy to show you around.